This is the final lab project for OPTI 380A. I'm Lindsey.
And I'm Bamberg, and our TA is Yoon Woo.
The goal of our project was to use spectroscopy to demonstrate the use of optical imaging to identify the difference between healthy and diseased tissue. Spectroscopy is the study and analysis of the interaction between matter and electromagnetic radiation. Materials have many different absorptive and transmissive properties which can be measured using spectroscopy. A material's spectral information can be found by passing light through a material and dispersing the transmitted light spatially into its constituent wavelengths, then mapping spatial positions to wavelengths. Current research at the University of Arizona includes using optical and spectral properties for a variety of biomedical imaging purposes, including diagnosis and surgical guidance. For example, research using the property of fluorescence is currently being conducted for both of these applications. Additionally, research is being done on absorption properties of tissues for cancer screening, and our project aims to replicate this.
To achieve this, we built a spectrometer from scratch and used it to measure the absorption of a cancerous tissue sample. To construct the spectrometer, we placed a small circular aperture in front of a mounted white LED source. A 75mm achromat lens was used to collimate the light from the aperture into the sample. A liquid crystal diffraction grating then dispersed the light into its constituent wavelengths. After the light was dispersed, an 85mm achromat lens was used to focus the collimated beam back onto a Thorlabs detector. To take data, we first measured the emission spectrum of our white LED source. This served as a basis to find the absorption spectra of our samples. To do this, a horizontal line profile of the image of the diffracted light gave the spectrum, and some calculations were done to convert the x-axis from pixel number to wavelength. To verify the accuracy of our spectrometer, we measured the transmission and absorption spectra of Thorlabs colored microscope slides and compared our experimental results with the specifications given by the manufacturer. After inserting each slide into our system, a new image was taken, which was the transmission spectrum of the slide. The transmission of the different slides was then compared to the specifications given by Thorlabs. For example, the experimental transmission of the green slide can be compared to the manufacturer's data, and it can be seen that our spectrometer is fairly accurate. Finally, we inserted the human pancreatic cancer sample into the system and measured the absorption. The resulting transmission and absorption spectra are shown here. It should be noted that the irradiance incident on the detector was very low due to the thickness of the sample and scatter caused by its protective coating, so the exposure on the camera needed to be increased, which also increased the noise of the image.
Our system demonstrates a simple understanding of how visible light can offer insights into the composition of tissues by assessing their absorption and transmission properties. Changes in tissue composition due to diseases and pathological conditions can alter their optical properties. By knowing the absorption of a sample known to be healthy and comparing it to that of an unknown sample, any differences can be investigated further to potentially diagnose diseases. Additionally, if an absorption spectrum includes the wavelengths absorbed by early warning signals known as biomarkers, then an early diagnosis can increase the chance of treatment success. Our system offers high spectral resolution due to the liquid crystal diffraction grating, and high-resolution systems like this one can more accurately identify these kinds of biomarkers to help characterize tissue properties and identify diseases. Biomedical imaging is critical in advancing our understanding of health and disease within many organisms. Spectroscopy is a vital component in this field and plays a crucial role in discovering the intricate molecular structures within biological systems.